Hi guys, it is Monday, it's week 6, it's winter 2021, and it's Endocrinology Day. Off we go. Let's talk about Cushing's syndrome. Actually, we're going to lead up to Cushing's syndrome. Let's just talk a little bit more about glucocorticoids. We've kind of talked about them, so it should be pretty easy. What is the main function and when we say glucocorticoids, we're mainly talking about cortisol, and we've talked a lot about where that's released from, and the adrenal gland, the zona fasciculata, and one of the main functions is it makes sure that you always have enough blood sugar. We said that stress can suck blood sugar out of you, and if you don't have food around to replace that blood sugar, like if you're sleeping at night, how are you going to get the blood sugar? Well, cortisol can run, or cortisol can supply gluconeogenesis, which is the making of glucose, which is sugar. And it does so by protein metabolism mechanisms and fat metabolism mechanisms, and we'll get into all that here. It also has a very important function in handling stress, and again, stress sucks glucose out of your blood, and cortisol is really important for replacing that. Also has an important function with swelling and inflammation. It suppresses the immune system, as we'll see. Hypertension's a little more mysterious. We said that some of the newer research thinks that cortisol sensitizes norepinephrine receptors on the tunica media of arterioles, so sympathetically released norepinephrine can cause a vasoconstriction and oppose the effects of nitric oxide. And we said that in our last lecture that people without a lot of cortisol, that mechanism can be lost and the arterioles can be too wide, and that can drop your blood pressure. Also remember that cortisol can bind to MR receptors. Those are the receptors for who? Those are aldosterone's receptors. So it also binds to those as well. And usually it doesn't cause, we talked about its role in the kidney. I don't think we have talked about how that's handled in the kidney yet, have we? But we will talk a little about that here in a second. And the main glucocorticoid is cortisol, of course. Corticosterone is also released. Again, zona fasciculata for like the millionth time. We've said of the adrenal cortex, it's released. Its release is stimulated after ACTH binds to fasciculata cells. And cortisol is a member of the HPA axis, so hypothalamic pituitary adrenal axis. You remember the players? So who's the player of the hypothalamus? That would be CRH. Who's the player of the pituitary? That's ACTH. Who's the player of the adrenal cortex? That would be cortisol. That's the HPA axis. So cortisol exerts 95% of all glucocorticoid activities, and we said it does have a significant mineralocorticoid effect because, again, it can bind to the MR receptors. MR receptors are called mineralocorticoid receptors. The affinity for them isn't the greatest. However, the normal plasma concentrations of cortisol is about 2,000 times greater. So therefore, even though it doesn't bind as good to the MR receptors, it can still bind. Fun facts, there's a cortisol molecule for all you biochemistry people out there. How does it travel through the blood? About 90% of it is actually bound to corticosteroid binding globulin. 10% floats free through the bloodstream. The free form has the most biological activity. And it binds to, we said, glucocorticoid receptors. Those are its receptors. Specifically, it binds to the alpha glucocorticoid receptors. Sometimes they're called GR receptors. Sometimes they're called type 2 cortisol receptors. So there's some AKAs for those. They are not on any cell membranes, because cortisol is a steroid. It can go right through cell membrane, no problem. The receptors live in the cytosol and are not on the cell membrane. And the binding of these receptors and cortisol, together they go into the nucleus and they target their genes, and the central dogma is invoked by this complex going in. So you definitely need, you can't get, free cortisol can't go into a cell membrane and manipulate genes. It has to have the GR receptors or the alpha glucocorticoid receptors in order to do this. And there are heat shock proteins. You probably have studied these before. They cover the binding sites of the alpha glucocorticoid receptors, and they're removed. We won't get into how they're removed, but that's so other things have a little more trouble binding with the GR receptors because of those heat shock proteins. And there's some mutations and problems with those, but that's for biochemistry, not me. Also noteworthy is the beta glucocorticoid receptors are not bound by cortisol. Cortisol in the kidney, and I think we've covered this before, but well, if cortisol acts like aldosterone and combined to aldosterone's receptors, can it bind to principal cells in the kidney? And can it overdrive the principal cells, act like hyperaldosteronism? And the answer is no. Normally it can't do that. And why can't it do that? Because there is a very important enzyme that floats around in the kidney, and it's called 11-beta hydroxysteroid dehydrogenase type 2. We'll just call it 11-beta HSD2. And that is super, super important. What does it do? Well, it grabs cortisol that comes into the kidney and comes near the principal cells. It grabs it and it turns it into a different molecule. In fact, it turns it into something called cortisone. And cortisone is much less biologically active, especially in the kidney. It doesn't do anything in the kidney. It can't stimulate the principal cells the way aldosterone does. So normally, cortisone can't cause the uptake of salt and water and the kicking out of hydrogen ion and potassium ion like aldosterone does. So that's a good thing. Well, so that, of course, is going to lead to a disease. So what if we get a mutation of our 11-beta HSD2 gene? That ain't a good thing. In fact, that leads us to this disease called AMES. So it's apparent mineralocorticoid excess syndrome, commonly known as AMES. So these patients have a mutated gene for 11-beta HSD2, and therefore when cortisol comes near the principal cell, tries to go after the MR receptor, it's not converted into it's not converted into cortisone, and it stays alive and it can bind to the principal cells. And therefore you have a situation where you basically have hyperaldosteronism, and we've already discussed that. Remember, that means that the principal cells take in too much salt and water, and your blood pressure goes way up because your blood volume goes way up, kicks out potassium and hydrogen ion. So you get alkalosis, you get hypokalemia. Yep, so it's not good. How does an AMES patient present? Well, I just said they have hypertension, they have hypokalemia, they have alkalosis, metabolic alkalosis. But here's the key. They have low renin as well because they have hypertension, so that's going to shut off the R2A system. But here's the key. They will have normal or maybe even low levels of aldosterone. So how can it be hyperaldosteronism when you don't have high levels of aldosterone? So that is the secret right there of diagnosing AMES. And I should probably have more stars here because I do like that question. Got it? Okay, what about licorice? There was actually someone who died of licorice who was eating so much licorice. So let's tell you the licorice story. There is a licorice-induced AMES. So people who eat tons of, and you have to be ridiculous amounts every day of it. But licorice has a biomolecule in it. Licorice can be converted into something called glycyrrhetinnic acid. Glycyrrhetinnic acid. And that's not good, because what does glycyrrhetinnic acid do? It blocks the enzymatic activity of 11-beta-HSD2. So that's not good, right? With this receptor blocked, renal cortisol will not be converted into cortisone. And you'll have the real deal. You have cortisone binding to principal cells and way overstimulates the principal cells. So that's not good. The hypokalemia can put somebody into cardiac arrest. The hypertension will not kill the patient, but it's the hypokalemia that can be very, very dangerous. Right, here's the article. This was in September of 2020, the New York Times article here. Man dies after eating a bag of licorice each day. And that's exactly what happened. All right, so more about the glucocorticoids. We've talked about cortisol. That's the principal glucocorticoid. Super potent. There's also corticosterone. That's in there. It's less potent than cortisol, but that's also released from the adrenal cortex. Only accounts for about 4% of glucocorticoid activity. What's weird about corticosterone, we have to go back to biochemistry. So here's the pathway to make aldosterone, and strangely enough, corticosterone lives in the pathway to make aldosterone. So that's just plain weird, right? And I don't have to go through this again because you guys probably have this memorized, but we have cholesterol and pregnenolone to progesterone. Progesterone can be split two ways to make cortisol and aldosterone, but on the way, corticosterone is one step away. Aldosterone synthase actually takes corticosterone and converts it into aldosterone. And remember, of course, how can you forget 21-hydroxylase, because we've already talked about it, 21-hydroxylase deficiency. All right, there are some more glucocorticoids, though. There's cortisone. We just met cortisone. That's 11-beta-HSD2 converts cortisone into cortisone. Just talked about that one. The rest of these are all synthetic, so you can take these as pills. Methylprednisolone, about 5 times as potent as cortisone. Look at this one. Dexamethasone is 30 times as potent as cortisol. So that's a really, really powerful one. It's long acting. And then hydrocortisone is less potent than the other ones. It's another synthetic glucocorticoid. This is the one that we talked about in adrenal insufficiency. That's commonly used to control. It's very short acting. These synthetic glucocorticoids were been used for a long time. Back in the 1949 was the first report of its not successful, but helpful treatment of rheumatoid arthritis. I mean, you can't fix rheumatoid arthritis, just prevent the inflammatory damage. But now it's used for all sorts of these synthetic glucocorticoids are used for all sorts of inflammatory conditions, including sciatica. And prednisone is also a very popular one. We've talked quite a bit about it. How do glucocorticoids knock out inflammation? We've talked a lot. Was that in GIGU, I think? We talked about the inflammatory pathway. But let's get a little deeper into inflammation. There's genomic and non-genomic mechanisms where these glucocorticoids can knock out the inflammation process. So genomic effects of glucocorticoids, they can turn off genes in inflammatory cells like macrophage T lymphocytes, natural killer cells. And they turn off these genes so they stop producing pro-inflammatory cytokines like the interleukins 1, 2, 6, and 8, tumor necrosis factor alpha. So that's one thing it can do. It can actually bind to receptors on these cells so they stop calling in the troops, right? Macrophage releases like tumor necrosis factor alpha like crazy. And that's like gasoline, and it calls in the troops. Now it also binds glucocorticoids like prednisone. It binds to phospholipase A2 and inhibits it from making arachidonic acid. So remember this mechanism. Remember tissue injury, phospholipids are released, and they're converted by phospholipase A2 into arachidonic acid. Steroids bind to phospholipase A2 and stop this. So steroids snip this way here, up here high in the bud. We talked about COX-1 and COX-2 inhibitors and their effect. But this is the ultimate. These like prednisone and all of the gang is the ultimate steroid because it can stop inflammation at the root level. But then again, it can cause all the same sequelae that we have problems with, like it can knock out your gastric mucosal barrier. So that's not good. It can cause anticoagulation. It decreases anticoagulation, and that's not good. So it could do some bad things as well. More genomic effects. So it also turns on genes in the T helper, T dendritic cells that make anti-inflammatory cytokines. So these are the good guys: interleukin 10, lipocortin 1, NF-κB inhibitor. So that's another anti-inflammatory type of. All right, cortisol versus inflammation and other mechanisms. So cortisol also stabilizes and toughens lysosomal membranes. So remember back from probably first quarter, this is one of the most, Guyton thinks it's one of the most important anti-inflammatory functions of the glucocorticoids. Because in the inflammation process, lysosomes are destroyed and they release some really dangerous destructive substances, because lysosomes are like little stomachs, right? They destroy just about anything, but if they explode and release all this stuff, that's not good. So cortisol kind of shelters them and protects them from doing that. Other mechanisms. They also cause decreased permeability of capillaries. So proteolytic enzymes released from the exploding lysosomes can increase capillary permeability. And that's not good because that causes blood fluid to leak into the interstitium and causes swelling. And so cortisol prevents that, or decreases permeability of capillaries. In addition, cortisol, we said, it stops these lysosomes from blowing up and releasing nasty stuff. Right? Attenuates fever. It also knocks out interleukin 1. Interleukin 1 stimulates the hypothalamic temperature centers and causes fever. And cortisol blocks that mechanism as well. More adverse effects with glucocorticoids. So like COX-1, as we just kind of said this already, they will knock out PGNE2. Right? So if we snuff this out here, I mean, this whole pathway is gone. So there goes your gastric mucosal barrier. PGNE2 won't be created. It'll be interfered with. And we need PGNE2 to make the components for the gastric mucosal barrier. I won't go through that lecture again. But that's important. So if you use glucocorticoids for rheumatoid arthritis or whatever, above physiological levels, so 7 millimeters, some people 5, but I mean, some people are up 20, 30, 40 milligrams of this stuff. If you use anything above physiological levels, though, you can start getting syndromes or effects of having too much cortisol around all the time. And that results in something called Cushing's syndrome. It's a Cushing's syndrome-like thing. It's caused from exogenous glucocorticoids, but nevertheless, you can get Cushing's syndrome type effects from long-term use of glucocorticoids. Also, coming off the glucocorticoids, we talked in depth about adrenal insufficiency, right? Because all your machinery is shut down when you come off. and that can be tough to get your machinery working again. Fun facts, most glucocorticoids have both glucocorticoid and mineralocorticoid activity. Hydrocortisone in particular has a lot of mineralocorticoid activity. The exception here is dexamethasone, the strongest glucocorticoid, remember that one? But it has only, or the strongest glucocorticoid, but it has almost zero mineralocorticoid activity. That's kind of a weird one. What about stress? We've talked about this in the past, I believe, but one key function is to make sure, and we've said this already, to make sure that you have adequate levels of blood glucose. If a tiger jumps out of the woods and chases you, you're going to burn up all your blood sugar and glycogen really, really quick. And you're not going to be eating in this stressful situation, so how do you keep glucose in your blood? Well, one important way is the effects of glucocorticoids. Cortisol will rip up your muscles and make protein out of them, and the protein can be run through gluconeogenesis. And it can burn up the fat, and you can use the glycerol to run through the Krebs cycle, which then can be run through gluconeogenesis to make glucose. What are some stress events? We've covered these before. Major trauma, infection, chronic exposure, surgery, severe illness, chronic confinement can do it, psychological conditions and stressors. And we said already, stress sucks up the glucose and glycogen like crazy, and it robs the food-generated blood glucose source. So where's the glucose? You're going to go into hypoglycemic shock. What's the body going to do? It's going to make glucose through the process of gluconeogenesis. Remember, some cells in the body they have to have glucose all the time. Brain cells and red blood cells, to name a few. They have to have glucose around or they will die. Cortisol feeds gluconeogenic pathway. Yes, cortisol not only stimulates gluconeogenesis, it also makes sure there are plenty of biomolecules around to feed this pathway. So we've said already, it breaks down fat into glycerol, breaks down muscle proteins into amino acids, and they can be used to be put through gluconeogenesis. And cortisol also, people with chronic prolonged stress, they can have chronic high levels of cortisol because of that, and that can lead to stress-induced hyperglycemia if those levels are super high all the time, and that can cause all sorts of trouble and Cushing syndrome like things, and can rip up your muscles and rip up your heart and rip up your liver. It's not good to have high levels of cortisol. There's a study, for example, there's a study where people in the intensive care unit, for prolonged periods of time, about 33% of them actually have hyperglycemia because of chronic stress-induced cortisol production. 33% of them, if they're in there for a long time or they're debilitated for a long time, or 33% of them go on to becoming full-blown diabetics within 12 months. These are critically ill patients. So, what is gluconeogenesis? Well, it's a biochemical pathway that makes glucose from non-food precursors, like, again, amino acids, glycerol, lactate. And we'll get into this more in the next lecture, but amino acids, but leucine and lysine don't play the game. You can't use them to make, that's a great board question, right? Leucine and lysine. You can't use them to make glucose. You can't run those through gluconeogenesis, which is strange. Alanine is one of the favorites, really easy to make glucose out of. Yep, so that's the story there. Where does it occur? Well, the liver, you probably know that. 90% of it occurs in the liver. The kidneys actually run a little bit of it. If you are fasting, starving, the kidneys, that ramps way up. The liver actually drops a little bit and the kidneys ramp up. Even the small intestines, the wall of the small intestine, some of those cells can run a little bit of gluconeogenesis as well. How does cortisol drive gluconeogenesis? Again, it breaks down fats and muscles to fatty acids and glycerol. Another note here, fatty acids cannot be used to make glucose. Okay, that's like leucine and lysine cannot. Fatty acids can't. So when you break down fat, fatty acids aren't used. Muscle proteins again, except for leucine and lysine. Could probably get rid of that slide. How else does cortisol drive gluconeogenesis? So it also stimulates the central dogma in cells to manufacture enzymes for the Krebs cycle. So you have an overabundance of enzymes. That way Krebs cycle can run at full blast without any limitations. And we'll talk about oxaloacetate as the main product. That's the goal of the Krebs cycle, is to make oxaloacetate, and that's what's run through gluconeogenesis. What else? Well, it stimulates not only Krebs cycle enzymes, cortisol can also stimulate cells to produce the gluconeogenic enzymes, so all the enzymes that run gluconeogenesis. So the Krebs cycle and gluconeogenesis, that has plenty of enzymes, so it never runs out. Why? Because of cortisol. All right, so I hope you remember your biochemistry and a little bit about gluconeogenesis and glycolysis in the Krebs cycle, because that's where we're going the next lecture. See y'all later.